here. Hopefully you can. So basically what we're starting off with is this series here, which is quite a small thing, and I really don't think you'll be able to see any of this. Hopefully that can focus, but I'm not sure if it will. Okay, let's see if I can get it to focus. There. Okay, so we are starting with up here we have our glucose, and it's going to go through the whole glycolysis to give us pyruvate, and then it obviously enters the citric acid cycle. Now here's the thing, fatty acid synthesis is only really occurring when there is too much glucose present in the blood, right? Maybe you're eating too much, or there is too much ATP being produced, which would also happen when there's high glucose, and of course with high glucose would mean high insulin release. So all of these things really are associated with eating too much or having too much of this energy, aka eating too much. And so what will happen is we don't want to have too much ATP being produced, and to maintain homeostasis we're going to prevent or inhibit this ATP production. And how we can do that is by this excess glucose being converted into excess pyruvate. Excess pyruvate will make too much citrate, and too much citrate will actually inhibit the rest of this citric acid cycle. And what happens is the citrate can leave the mitochondria, and it can be converted into oxaloacetate and acetyl-CoA, and this is done by citrate lyase here. And what this will do is we are going to be producing oxaloacetate, and this citrate lyase is going to be bringing in coenzyme A. So we can basically do the reverse of the citric acid cycle, sort of. So we can produce our, so we're combining the coenzyme A with oxaloacetate to produce malate, and then via the help of an enzyme called malic enzyme, we have reduction of NADP. By taking hydrides to give us NADPH, which we'll use later on. And so this malate can be converted back into pyruvate, where it can of course enter the cycle once more, the citric acid cycle. Now here we said that we were breaking down the oxaloacetate into our the citrate into oxaloacetate and acetyl-CoA. And so the acetyl-CoA, if you remember, is a 2-carbon compound. So this is going to be acted on by acetyl-CoA carboxylase, which has been biotinylated, and this will allow for the addition of a carbon dioxide molecule to this 2-carbon acetyl-CoA to produce our fatty acid precursor known as malonyl-CoA. And so now we can actually get started with it. So I've drawn these two coloured transporters here, and on the very outer portion of the mitochondria, what we have is this pink dot is supposed to represent carnitine A cell transferase type 1, which usually brings fatty acids into the mitochondria for beta oxidation, and the blue one, which is on the very inner portion of the mitochondrial membrane, which would be bringing it into the matrix, is carnitine A cell transferase. Remember, both of these can be referred to as carnitine A cell palmitol transferase. So the pink one is type 1 and the blue one is type 2. Now, with excessive amounts of malonyl-CoA, what we have is the inhibition of this carnitine A cell transferase type 1 carriers. So we are preventing fatty acids from actually being brought into the mitochondria. So we're inhibiting beta oxidation. And so this means that what we'll end up with is fatty acid synthesis instead of the beta oxidation that can occur. Now, here you can see I'm starting to explain this fatty acid synthesis. So what happens is we have the acetyl-CoA from up here is going to be acted on by acetyl transacylase here. So we're removing the coenzyme A group, so we only really have our acetyl group that we're interested in from the acetyl-CoA, and this is going to be added on to a specific enzyme called fatty acid synthetase type 1. Now, it has two important domains that we're going to be focusing on, a cysteine domain and our ACP domain, which is our A cell carrier protein. And the acetyl group from the acetyl-CoA is going to be transferred onto the ACP domain of this enzyme. And this, we can, instead of referring it to acetyl, we can call it an acetate group. So the acetate is being added to the ACP, and we just have that removal here of the CoA from the acetyl-CoA. Then what happens is we're going to have that acetate is going to be removed from the ACP domain onto the cysteine domain of the specific enzyme, which can bring us on to our next step on this piece of paper here. So we've now removed the acetate from the ACP end, and we've added it onto the cysteine domain. Now, we also have this thing called myelinate. Where does that come from? Well, malonyl-CoA, if you remember, we said was basically a fatty acid precursor. What happens here is we have that malonyl-CoA, we have the removal of CoA, and that's done from the malonyl-CoA, and that's done by malonyl transacylase. And so we're really only adding our malonyl group. And if you remember, when we were adding our acetyl group, we only referred to that being as adding an acetate. And so in this case, we can say the same thing. Instead of a malonyl group being added, we can call it myelinate. And so the myelinate is being added to the ACP end of this particular enzyme. Then what happens is, by the help of malonyl acyl ACP condensing enzyme, we have a decarboxylation reaction occurring. And now specifically at the myelinate group. And so this is a 3-carbon, but following the decarboxylation, it's obviously going to be a 2-carbon. And I'm going to refer to this continuously throughout my diagrams as myelinate, but it's actually a 2-carbon myelinate breakdown product. So then what happens is we are going to actually have a condensation reaction between this myelinate here, which has undergone decarboxylation. So now we have our 2-carbon breakdown product, which would be on the ACP. However, we are going to actually have a condensation reaction. So we're moving it from the ACP domain back onto the cysteine of this enzyme again. And so we have our 2-carbon acetate, and we have now a 2-carbon myelinate breakdown product, which we can refer to now, as these have been condensed, as a 4-carbon butyl group. So next up, we have, by the help of beta keto acyl ACP reductase, we are using this enzyme to actually convert NADP into NADPH. So that means that this itself has been oxidized because we are depositing these hydride ions onto the butyl group. So we are having the conversion of a ketone into an alcohol. Now, following this, we are going to have a dehydration reaction, as you can see here, which is done by our beta-hydroxyacyl ACP dehydratase. And this is going to, of course, is a dehydration reaction, so we're having a double bond forming within that butyl group. Now we're almost at the end of this fatty acid synthesis of the first stage of this, really. So we have a double bond within this butyl group now, and following this double bond production, we have enol-CoA ACP reductase, which is going to actually drop hydrides across that double bond within that butyl group here. So now we're going to be breaking that double bond once more to give us a single, revert it back to our single bond butyl group attached to this enzyme. And then that's being mediated by, again, the oxidation of NADPH to give us our NADP+. So now we're at this final step where actually we have malonyl transacylase coming back and adding our myelinate onto the ACP and removing the CoA from malonyl-CoA. So hopefully you can see now that we have our specific this structure, which is our butyl group, which contains no double bonds now, attached to the cysteine of this particular enzyme. And ACP, this ACP domain now also has a myelinate. So this has 3 carbons attached to the ACP at the end of it. And this cysteine portion of the enzyme has 4 carbons attached to it. And then what can happen is this can restart, because what did we say? Well, we have myelinate here. We have myelinate here. Where else did we have myelinate attached to ACP? At the very near the very beginning of this fatty acid synthesis, except now instead of the cysteine residues only having acetate attached to it, it had that acetate and myelinate breakdown product. So this already has 4 carbons attached to it. Now this has 3 carbons attached to it. So then what do? We are adding another. Then what we can do is continue this process. We have decarboxylation, and then we are adding the myelinate from here back onto here. But this already has 4 carbons. So when we are doing the decarboxylation step and we're transferring the myelinate onto this portion of the enzyme, we are going to have, in fact, a total of 6 carbons here. And this actually continues until we have at least a minimum of 16 carbon fatty acid produced.